Good afternoon, dear students. Today we are going to discuss about production of the antibiotic penicillin. Now, the antibiotic properties and the possible therapeutic value of penicillin was first discovered by Sir Alexander Fleming in the year 1929. What he observed was that the growth of Staphylococcus aureus on agar medium was inhibited by a fungal colony. Right. That means around the fungal colony there was no growth of Staphylococcus aureus. Later on, after identification, the fungus was identified as Penicillium notatum. A soluble and a diffusible product was produced by the mold which had inhibitor activity. That means a soluble product, a diffusible product was produced by the mold Penicillium notatum, which had the ability to inhibit the growth of S. aureus. This material was called as penicillin after the name of the mold which produced it, that is Penicillium notatum. The penicillin that was observed by Fleming was penicillin F. Okay. Now, penicillin was known as magic drug during the World War II. It was very active against gram-positive bacteria, nocardia and actinomycetes. But it was not active against most of the gram-negative organisms, except when it was used in high-level doses. Now, how does penicillin kill the organism or how it affects the gram-positive bacteria? It kills the cell by degrading and inhibiting the cell wall synthesis. It degrades the cell wall or it inhibits cell wall synthesis. However, penicillin is non-toxic to mammals except in certain individuals who can get allergic reactions, and that too because of the side chain. Okay. Now, let us discuss in detail about penicillin production. Number one, so while studying penicillin production, the first thing is the mold, the fungus that is used for penicillin production. The mold that is preferably used for penicillin production is Penicillium chrysogenum Q176 strain. Okay, which strain is used? Penicillium chrysogenum Q176. This strain is a mutant strain. It is actually a mutated organism obtained by treatment of the ancestral strain Penicillium chrysogenum NRRL 1951. Okay, so Penicillium chrysogenum Q176 strain used for penicillin production is a mutated organism, okay, obtained from Penicillium chrysogenum NRRL 1951. Okay, and how it is mutated? By treating it with mutagenic agents like X-rays, UV light, or nitrogen mustard gas, right? Now, Q176 is a high yielding strain. Okay, so after repeated cycles, after multiple cycles of exposure to X-rays or UV rays, Q176 strain was obtained. It's a very high yielding strain. Now, this production strain is stored in the dormant form as spore suspension. Okay, so in the you can say laboratories or culture collection centers, it is stored in the form of spore suspension. How it is stored? In the form of a spore suspension mixed with sterile soil or sand and desiccated. It may be stored in by lyophilizing in appropriate media. It can be stored under liquid nitrogen in frozen state. Okay, so it is stored as spore suspension either by suspending in soil or sterile soil or sand, or it may be lyophilized in a particular medium, or it is stored under liquid nitrogen. Now, during the production, we use two types of media, right? Number one, sporulation medium, which is required for inoculum development, and number two, production medium. Okay, so we use two types of media. Now let us study inoculum preparation. For inoculum production, the spores from stock cultures are propagated on a special type of media called as sporulation media. The sporulation media contains glycerol, cane molasses, constip liquor, magnesium sulfate, potassium dihydrogen phosphate, peptone, sodium chloride, iron tartarate, copper sulfate, and distilled water to make the medium. Agar-agar is added for solidification if you require. Now, the medium is seeded and incubated. Okay. If we do not require a solid medium, we need not add agar-agar. After seeding, the medium is incubated on a rotary shaker for aeration and agitation. Okay. So, you have a sporulation medium inoculated with the spores from the stock culture and incubate on a rotary shaker or shaking incubator for continuous aeration and agitation. The solid media are supplied with adequate air supply. The temperature is controlled at 24 degrees Celsius. Then the spores are transferred into the next stage. Okay. So inoculum development, stepwise increasing in the volume of the inoculum. The volume of the culture increases 10 folds in each successive stages in the progression of inoculum preparation. Okay, so from one stage you transfer it to another stage and so on and go on developing the inoculum, and at every stage there is 10-fold increase in volume. This is how we prepare the inoculum by using sporulation medium. And then for production of the antibiotic we require a production medium, right? So first you have laboratory stages and then production stages, right? A specific type of medium called production medium is used for penicillin manufacturing. The production medium contains CSL, that is corn steep liquor, glucose, calcium carbonate, potassium dihydrogen phosphate, and vegetable oils. Penicillin precursors may also be added depending upon the type of penicillin to be produced. Okay, precursors are added but the type of precursor added depends upon what type of penicillin you want to manufacture. Now, what is the role of various ingredients or raw materials during penicillin production? First is carbon source. Lactose is a better carbon source than sucrose and glucose because lactose breaks down slowly into glucose and galactose. Thus, there is a steady but not abundant supply of carbon, right? So the production medium, the production medium which we use contains what? Lactose sugar, constip liquor, glucose, calcium carbonate, KH2PO4 and vegetable oils plus precursors. Right. Why it contains lactose also in addition to glucose? Lactose is a better carbon source than sucrose and glucose because lactose breaks down slowly into glucose and galactose, and there is a steady but not abundant supply of carbon. So slow supply of carbon is there. Glycerol and sorbitol are less suitable, so they are not used. The next is nitrogen source. Corn steep liquor present in the medium is a cheap, readily available nitrogen source. Instead of CSL, we can use ammonium sulfate or ammonium acetate or ammonium lactate or even ammonia gas as a nitrogen source, but CSL is more than enough. Corn steep liquor is sufficient. Then minerals. Okay, elements like potassium, sulfur, phosphorus, magnesium, zinc, and copper are required by the organism, right, or by required during the production. Now, corn steep liquor, which is present in the medium, supplies some of them. Now, media containing CSL, corn steep liquor, does not require magnesium or zinc ions. Otherwise, they can be supplied as MgSO4 as their sulfates. Okay. Now, KH2PO4 supplies potassium and phosphorus. Iron and copper are supplied as sulfates if required. Iron in the form, copper in the form of copper sulfate and like that. Okay. Now the KH2PO4 that is present in the medium also serves as a buffering agent. Potassium dihydrogen phosphate also serves as a buffering agent. Calcium carbonate present in the medium also helps in stabilizing the pH. The vegetable oils that are added into the production medium serve as antifoam agents. Oils, being fatty substances, also supply energy to the microorganisms. Now, precursors. Corn steep liquor contains phenylethylamine Constiped liquor contains phenylethylamine, which supplies phenylacetic acid, which serves as a precursor for the synthesis of penicillin G. Okay, so when you use constiped liquor, okay, it contains phenylethylamine, which supplies phenylacetic acid, which serves as a precursor for the synthesis of penicillin G. Phenylacetic acid supplies the side chain of penicillin G, that is benzyl penicillin, right? So this is about the sporulation medium, inoculum development, and the production medium. Now, actual fermentation. The fermenter used generally is of glass. It has a capacity of 5,000 to 10,000 gallons. Okay, during the course of fermentation, you have to maintain the pH at 4.5 to 5.5 and maintain the temperature at 24 to 25 degree Celsius. The duration of fermentation is around 5 days. The entire fermentation process can be studied under 3 phases. Number 1, growth phase. Number 2, maturation phase. And number 3, decline phase. What happens during growth phase? During growth phase, the spores germinate and multiplication follows. That means sporulation and multiplication stage. Here in the growth phase, the spores present in the medium, in the inoculum, develop, germinate, and they develop into mycelium, and then the multiplication takes place. Okay, spore germination and multiplication. The dry spores suspended in a non-toxic wetting agent like sodium lauryl sulfonate, which is 1:10,000 diluted. Okay, so the dry spores are suspended in a non-toxic wetting agent or simply in sterile water, and it is applied into the fermentation medium, into the production medium. The spores are applied using spray guns or pipettes, and then the medium is aerated and agitated for equal distribution of spore suspension. The fermentation medium may otherwise be inoculated using pellet inocula also. The spores germinate and the vegetative cells or the mycelia that develop undergo multiplication, or the vegetative cells undergo multiplication to form mycelial mass. The organism rapidly uses glucose, which is present in the medium, as a carbon source during growth phase. Okay, so the first phase, when you inoculate the spore suspension into the production medium, perform aeration and all and agitation, then during this phase the glucose that is present in the medium is readily utilized as a carbon source by the organism for development of mycelial mass, rather you can say for growth and development. Okay, leading to mycelial mass development. But during this phase, very little penicillin production takes place, or rather you can say penicillin production is not initiated during the growth phase. Now this growth phase is completed within 1 to 2 days, that is within 24 to 48 hours. Okay, once after this, in the second phase, which we call as maturation phase, since now glucose is exhausted, the organism uses lactose as a carbon source. But actually, lactose is a disaccharide and it is not readily available to the organism. And hence, when glucose is exhausted, when glucose is present in the medium is exhausted, finished, the mold is being starved at this point. Organism undergoes starvation because there is a lag phase and the organism undergoes starvation, and very little further growth occurs. Once the organism undergoes starvation, upon exhaustion of glucose, very little growth occurs. Now the organism shifts to lactose. Now, lactose being a disaccharide is slowly degraded into glucose and galactose. And slow availability of glucose allows starvation condition that is actually required for penicillin production. The mold produces maximum penicillin during starvation period, and starvation period is completed within 5 days. Okay, so maximum growth occurs in 1 to 2 days when glucose is available. As soon as organism shifts to lactose as a carbon source, there occurs a starvation period and the mold starts producing penicillin. Okay, this phase is completed in 5 days. But at the end of fermentation, the pH rises to 8 or higher, since depletion of lactose brings about autolysis of the mycelium. This is called as decline phase because during the exhaustion of depletion of depletion of lactose occurs, pH rises to 8 or more, which brings about the autolysis or lysis of mycelium, and decline phase. The organism starts dying. During this phase, the penicillin produced may also be degraded, and hence the fermented broth is harvested before the decline phase begins. Okay, before the autolysis of mycelia begins, fermented broth is harvested. Okay, so that penicillin is not affected. So from this graph you can see that after a period of 2 days, after a period of 48 hours, when the growth phase is over, the penicillin production actually gears up. Okay, the lactose gets exhausted and penicillin production starts only after growth phase. Now, in this flow sheet, you can see you have a stock culture of Penicillium chrysogenum Q176, which is subjected to inoculum development in a shake flask and then subsequent transfers, and then into a seed tank having sporulation medium. Finally, you transfer the inoculum into a production tank, that is fermentation vessel, which contains production medium, where the pH is maintained at 4.5 to 5.5, temperature at 24 to 25 degrees Celsius, and a period of 5 days or a week may be required. Then once the fermentation is over, you go for harvest and recovery, where the fermented broth is passed through a filter press. That is, you can say it is filtered and then extracted out in aqueous phase and organic phase. So there is a shift between extraction of the compound in aqueous and organic phase. We'll discuss it later. Then from the first extraction, then second extraction, and then third countercurrent solvent extraction process. Okay, so first time, second time, third time extraction is done, and finally the product is crystallized. The crystals are washed. Okay, and then you add NaOH or KOH to get sodium or potassium salt of penicillin. The metal salts of penicillin are subjected to purity potency testing. It may be filled into vials or converted into tablets, sealed, labeled, and then transported. Okay, so this is how production is done. Right, so let us discuss about harvest and recovery of the product. Penicillin produced by mold is present in fermented broth. That means it is present in aqueous phase, and it has to be separated out from the mycelium. You have to remove the mycelial mass and other impurities. So the fermented broth is filtered by using rotary vacuum filter or filter press. Now the filtrate contains penicillin, and the mycelial mass and other solids can be pressed and used as animal feed. The next step is extraction of penicillin by countercurrent solvent extraction process. For that, the aqueous filtrate is the pH of the aqueous filtrate is lowered by adding sulfuric acid or phosphoric acid. The pH is lowered to 2 to 2.5. Okay, so the aqueous filtrate is mixed with sulfuric acid to reduce the pH to 2 to 2.5. This converts penicillin into anionic form. This anionic aqueous penicillin is immediately extracted into extracted with an organic solvent. You mix it with organic solvent like amyl acetate or butyl acetate or methyl isobutyl ketone in a countercurrent solvent extractor. So that means it is extracted now in organic phase. Okay, then the penicillin is extracted back into again aqueous phase by adding enough of KOH or NOH to form salts of penicillin. Right, so first you have aqueous penicillin in aqueous form, you extract it into organic phase, then you go back to aqueous phase, then again organic phase. So okay, and then you add enough of NaOH or KOH to get salts of penicillin. Right, now this shift, the resulting aqueous solution is again acidified. Okay, the resulting aqueous solution is again acidified, re-extracted in organic solvent. So there is a shift between organic phase and aqueous phase, which helps in purification of penicillin. Finally, the solvent extract is carefully, or rather you can say the solvent extract is carefully extracted back with aqueous NaOH. Okay, when you extract it finally with NaOH, you get sodium salt of penicillin. This is followed by charcoal treatment. Okay, now you perform purification. Okay, so this is followed by treatment with charcoal to remove pyrogens. That means fever causing substances are removed. So removal of pyrogens which can cause fever. Then it is sterilized by using size filter to remove any bacterial contamination. Then finally, after purification and sterilization, you get pure metal salts of penicillin, that is sodium or potassium salt, depending upon whether you have added NaOH or KOH. So the metallic salts of penicillin can also be safely sterilized by dry heat if it is required. Then the aqueous solution of penicillin is subjected to crystallization. Okay, finally the crystals are again washed. Now the crystals can be packed, it can be lyophilized and packed as powder for reconstitution before use. It can be prepared as suspension and packed into bottles. It can be tableted for oral use also. During, you can say, the production, sterility, strict purity, potency testing is done. Freedom from pyrogens and sterility are performed. There should be no pyrogens, fever causing substances. It should be sterile, potency and purity is done. The final product must satisfy all strict government standards before sale into the market. Now this is the general structure of penicillin. You can say thiazolidine ring, you can see beta lactam ring. Okay, this basic moiety is 6 amino penicillinic acid. Okay, now this penicillin has a side chain. This is acyl group. Okay, it has a side chain of acyl group, right? Now this is a basic moiety which is 6 amino penicillinic acid, and it is having a side chain. With the change in, okay, the side chain can be different for different types of penicillin. So this is the basic structure of penicillin. Okay, now this is all about penicillin production. Now we have to discuss semi-synthetic penicillins, that we will discuss in next lecture.